you really need those conserved regions with all targeted organisms. And then, on the contrary, you need your barcode to be variable. It also, another thing that can be obvious is that your barcode needs to match something you're going to use. And as it must be in reference libraries, particularly if you are giving a taxation. First, the different parts of the cells. So for prokaryotes like bacteria, you will have only one DNA. That is quite easy. But for eukaryotes like animals and plants, you find DNA also in the nucleus, but also in the mitochondria and the chloroplast. And this, why is that? It's because if we come earlier in the evolution of a eukaryotic cell, it didn't have these organelles, this chloroplast and mitochondria, but in fact, this primary eukaryotic cell on the side an anaerobic, then a photosynthetic prokaryote that be more closer to DNA of prokaryotes than the DNA in the eukaryotes in mind. So famous and important marker genes are the genes coding for ribosomal proteins. They can theoretically investigate the global diversity in the tree of life. So the ribosomal genes, they code for the large and small ribosomes, which are responsible of RNA to proteins, as Agnès explained earlier. So it is illustrated figure on the right. And this is, of course, that's why you will find those genes in every organelle. And they are also present in chloroplast. Those ribosomal genes, many of them, that we code for the different units of the ribosome. For instance, eukaryotes, the small unit of the ribosome is coded by the 18S gene, while the large subunit is composed by a combination of other genes. These genes are slightly different for prokaryotes and also chloroplast and mitochondria, as the small unit is coded by the 16S gene and the large units of ribosome is coded by the 20 gene and the 5S gene. And what it is, and this organized in an operon, as I wrote, that means that they are clustered together in the genomes and always found in the same order. And why are they very interesting for metabarcoding is because within those genes, you will find an alternance of variable regions and very constant. So it's illustrated in this graphic where this study, they took 18S genes of a wide diversity of copepoda, and they aligned all those sequences. And then they look at each base, the entropy, that is the variability of the bases at this position. If you have a lot of different bases, T, C, G, A, C, G, etc., at a position, you will have a high entropy. But if you have almost the same base at this position, you will have a low entropy. And so we can see in this graphic that we have 9 hypervariable regions in the 18S, and this is true for all other organisms. Also, as they are organized in an operon, you can find internal transcript spacers that are present between 2 genes, And those regions are not coding. That's why they can be also hypervariable. They are hypervariable and can be used in a metabarcoding study as well. So it is then easy to design and find barcodes in the 18S, in the ribosomal genes. Something that you need to have in mind is that these ribosomal genes are very important for the biosynthetic capacity of the cell. And so for that, they are present in multiple copies. And this number of copies varies between species within a taxonomic group and also between taxonomic group. It is illustrated in this picture, where you have the number of copy of the 18S genes in different group of protists, flagellates, diatoms, dinoflagellates, and xylates. And you see that you can have a very different range in copy numbers for those species. So it's something to have in mind when it comes to analyze the abundance of those species, that it can influence the abundance at the end. Then there are other genes that can be used, for instance, for animals. And Agnes described these genes a little bit. It was historically used for the barcoding of insects. So it's a cytochrome-oxidase 1 gene that is present in the mitochondria, the key enzyme for respiration, and that is present in all animals, eukaryotes, and all eukaryotes. So when it comes to do metabarcoding, we already had a lot of barcodes and primers designed for those genes. So they are very interesting. They were very interesting to use. And they often provide a better taxonomic resolution than the nuclear 18S genes for animals, for some group of animals. But contrary to the ribosomal genes, it's quite complex to define universal primers. And I'm going to illustrate this with an example. Here you have the same graph I showed you earlier, but with 40 species of insects among 25 different orders. And we have the entropy at each position of the 18S gene. And we can see that we can define conserved regions that do not vary between all the different sequences we looked at. But when it comes for looking at the cytochrome oxidase genes, it's not the same. It's more difficult to find those conserved regions. So that's why it's difficult to use this gene for a survey of very diverse groups of animals. And last, genes are for specifically targeting photosynthetic organisms that are genes present in the chloroplast. So I won't get into detail more for those genes, as we will discuss them more into the next lessons. But something that you also needs to have in mind is that when you target chloroplast genes, you don't have the same number of chloroplasts between different phytoplanktonic cells. And but this can be used and this can be used as an advantage, and Frederic will show you that later. So the next step, the conserved region. I will get more into details how the primers are designed to match those conserved regions, and how you manage to have to target all the organisms you're interested in and not the other ones. So basically, primers are complementary sequences of the conserved regions. So you will have a forward primer that will hybrid to the conserved region that is at the beginning of the barcode, and you will have a reverse primer that will be complementary of the region you have at the end of the barcode. And they will be complementary and form hydrogen bond, as Agnes told us earlier. And for the specificity of the primer, it's basically to search for sequence that are only present in one particular group of organisms. And for that, you align different sequence of different organisms, and you look, okay, this sequence is only present in all the diatoms, so it's an interesting sequence to design the primers. And when it comes to investigate large, very large group, you can also use what is called the degenerescence levels of the primers. That is, you surely will have slight variation in the conserved regions for very large group. And so your primers, for instance, it's in the bottom right of the slides, you see that between the three different species, the first, the A at the third position is not present in the last species that has a G. So for this, for the design of these primers, you can put primers that will have either the complementary basis of A, that is a T, or the complementary base of G, that is a C. And it can be written a Y based on the code that is available online. And so with having degenerated primers, you can. And so depending on the groups you want to target, you will play with the specificity and this degenerescence of the primers. And I give you here an example of primers of the 16S ribosomal genes that are used to target the whole diversity of bacteria and archaea. And you see that there are two degenerated bases for the forward primers and 4 for the reverse primers. So then barcode needs to be variable in order to distinguish the different species you want to study. But as Agnès told us earlier, this can vary between taxa. And as I told you earlier, this can vary also depending on the barcode you're going to use. So it's also an important thing to have in mind when you need to choose your barcode. So another thing for your barcode is that it matches the sequencing to technology size. So here you have the broad size length of the different gene bear paste. And currently the sequencing technology allows you to have sequence of around 400 and 500 bases. So you cannot sequence the entire genes. And also for some metabarcoding studies, if you are interested in ancient DNA, your DNA will be highly degraded. So because you will only have short sequences. Again, you can have some drawbacks from this sequencing technology side. Here you have an example of primers, the global protease diversity. So it's a very large and diverse group, and have the super group. And you can see that the first primer that targets the hypervariable region 4 of the 18S ribosomal AS gene, it managed to get most of the super group. So it's you see that some are quite because you are limited by limited technologies. It's something that is going to improve in the next, can be a problem. So finally, the things to get from this is that the perfect barcode does not exist, and you have to select the one that will best suit to your research questions. If you are interested in invasive species detection, you will need a barcode with a very important variability that you will be able to distinguish precisely the species you're interested in. If you're more interested in global diversity and community dynamics, you won't be that interesting in the taxonomic assignation, but will be interested in that your primers can get you the largest diversity of organisms. So how do you select your barcodes? So 99.9% of the time, you will look into literature and see the ones that were previously designed. And then in that way, you can compare with other data sets. You know the biases of those primers. Okay, I won't have these kind of organisms, and you'll know that. And you know that it can work. For instance, the primers for diatom biomonitorings, you know that they can work for doing diatom biomonitorings. But if you are studying very exotic organisms, maybe those primers are not adapted to your study and consuming, and with a lot of optimization steps. And we will talk about this for the phytoplankton metabarcoding part, because it's what we try to do. And the last thing I wanted to discuss in this talk is the reference libraries that are very important, particularly if you want to have a taxonomic name for your organisms. So they are truly a prerequisite to identify your sequence and give it a taxonomy. It's as simple as if you don't have the sequence in the reference libraries, you won't be able to give it a name. So your reference library, you need it to be the most complete as possible. Here you have a study where they investigated microinvertebrates in the Mediterranean Sea, I think, and you have in green for different groups of microinvertebrates the sequence that could be identified, and in gray, the ones that couldn't be identified. So it is really an ongoing process to complete those reference databases, but it's a long process and it takes time. And also you want your reference databases, libraries to be the most curated as possible. That means that you have the true taxonomic identification of your sequence that you can rely on. So I will now present you an overview of some reference libraries you can find for the different marker genes I presented you. So for instance, for the ribosomal genes, one is generalist libraries where you will find a lot of sequences and more specialist reference of organisms. So for the ribosomal genes, one of the most famous database is SILVA, that really gather high quality sequence for bacteria, archaea, eukaryotes, and also for the mitochondria and chloroplastic genes. And then for those ribosomal genes, there is here too that gathers all the nuclear 18S, more than 200,000 nuclear 18S sequence for all the protists. And these databases has a taxonomic curation done by experts. So it is the same principle for phytoref, but that targets the 16S of chloroplast and to study phytoplankton. And here in the lab, we developed another reference library, and I won't get into detail because Benjamin will speak more about this later, tomorrow. For the other genes, I will mention bold databases. That is very important because you have more than 1 million 1 barcodes, and you can also have information, as Agnès explained, of the specimen collection, if this sequence comes from a collection specimen, the localization, where it was, where the sample was taken, etc. For more specialist reference libraries, you have diet barcode that we developed in the labs, specifically for the RBCL gene for diatoms, and Frédéric will talk about this later. And I'm not an expert of insects, but I found this in the internet. You have, for instance, these databases that can be used to study insects. In fact, depending on the groups, you might find maybe a reference library that will be more precise to your targeted organisms. And there is another type of libraries that are the nucleotide study DNA. Some, and you publish your results. Some journals may ask you to deposit your sequences in nucleotide databases. And there are 3 main nucleotide databases from Europe, ENA, Japan, DDBG, and NCBI from the US. And that comes from studies, things and DNA projects. For NCBI, it's more than 200 millions of sequences, but it relies on the taxonomy people gave at the beginning. So you are not errors. So you have it's interesting to take different reference libraries when you study your sequence and to combine the information you get. And the last things I want to say about reference libraries is that they are essential to find the best design for the primers. The more sequence you will have in your reference libraries, the more precise you will be in the design of the primers. If you want to target the whole phytoplankton and you only have diatoms sequences, your primers might really well target the diatoms, but not other groups of phytoplankton. So it's very important, and that's why we keep having more and more well-designed primers, the more sequences we have in the reference libraries. And it's also interesting to test for the specificity of your primers, that is, if they are not going to target unwanted taxa. And you have different tools that can be used, and for instance, Silva database has an interface, a web interface, where you can test your primers. So you have your sequence, and then it gives you, okay, your primers allow you to get 40% of this group of organisms, etc. So that's it for the first part. We saw the choice of the barcode and the reference libraries, and after lunch, we will see all the other steps. If you have any questions.
Thank you, Clarice, for this very nice and informative presentation. I have a lot of questions related to phytoplankton, but I think we will answer them during these days, especially at the day 3. But for now, can you tell us, when it comes to the phytoplankton, you said that there is no perfect barcode universal for phytoplankton. So can we target only cyanobacteria? Do we have universal and ideal barcode for cyanobacteria?
When I say there is no perfect barcode, is that you won't, or for now, and I believe it will be for forever, you won't have 100% of your phytoplankton diversity, but you can reach 80%, 85%. With some groups that will be missed, but 85% is still a very informative diversity that can be used. And for For instance, for phytoplankton, we managed in the lab to design a primer, and you can find them in literature as well, that target both cyanobacteria and eukaryotic phytoplankton. It's possible.
And you think it's possible to design or to use it for the biological quality elements?
I think Benjamin will answer better than me for this part, but yes, you will see. It will be a discussion of tomorrow. I won't spoil.
Thank you.
Yes, sorry. Just to, I would like to think about you, and I would like to be sure that you understand some graphs, which are, I think, important. And also, it's also about the skill evaluation of Friday that you will have to pass. No, but maybe, Clarisse, could you come back to, you showed this entropy graph, and to be sure people understood well, yes, this kind of graph. Maybe just before. Yes, this one. Do you understand? Everybody understand this graph, to be sure? Yeah. Not really. Okay. So, Clarisse, can you explain this again?
Yes. I hope I will be better this time.
Yeah, maybe understand step by step, but you have to align the sequences. That's okay.
So, for instance, in this study, they took the 18S sequence for 192 Copepoda species. So they had this almost 200 sequences, and oh yeah, at the end, yeah, an alignment is, yeah, it's basically you put all your sequence together and you find all the positions that are common. Yeah. Yeah, one line is a sequence of one organisms, and one each column is a base of this sequence. So in this graph, you will have an A for almost everyone, a T, then a C, then a C, and etc. And so to see what are the conserved regions and hypervariable regions, you just look if in this column where you have one base, if it's almost blue, that means the entropy is very low because you don't have so many different bases at this position of the gene. But when you look on the for the graph on the left, you will see, for instance, in this column that you have a much more different bases, and the entropy will be higher. And that's just a way to represent the variability of the sequence. And you do this all along the gene for each position. So here is the graph, it's for each position of the nuclear 18S, for instance. Yeah, and then yes, here, and then so here you have the entropy, and here you have the almost 2,000 bare pairs of the 18S, and you see that in this region, hypervariable region 2, you have very high entropy. That means the sequences are really different between all the species you analyzed. But when you look at this, the entropy is much more lower, and that is a conserved region. And it's what you want for your primers. Yes.
I just want to see, did I understand this well? So, for example, we have 10 diatom species. Achnanthidium, Minotissimum, Cimbella, and so on. And this region, which is conservative between these species, means that they have at the same position the same order of nucleotides. And we can use it because there's not change among species.
Perfectly.
In the between of the two conserved regions, you have this hypervariable region where which is highly diverse among your species, so that may help you to really distinguish between the species, because each sequence will be different.
And it will be different also among specimens in the same species.
Sometimes.
Sometimes.